and followed by we'll discuss some of the results, uh, the fantastic results we gained in the research outputs. The title is known as an volatilization of coal fly ash into sodium silicate nanoparticles for salinity stress amelioration in rice. Why we are choosing these kind of specific aspect? Like in India, we are belongs to most of the energy generation towards the some kind of like thermal products only. In in India itself, several thermal power stations will be available. I belongs to a southern Indian part. In southern Indian part, one of the familiarized uh, thermal power stations, which is located in the Thuthukudi, which is uh, in the region of Bay of Bengal, in between the join the below the above around 500 kilometers of Indian Ocean. Like specifically, we have collected why these specific coal ash means these coal ash will be going to the contamination into the soil and give the pollution towards the air and chemical, which decreases the agricultural productivity. According to the United States Food and Department of Agriculture, over 5 million polluted sites are globally converting approximately 20 million hectares of contaminated lands, soil deteriorations, which are major agricultural challenges. And also, this is one side, and one side, the increasing growth of urbanization. Okay, most of the lands, most of the agricultural lands that will be going to contaminated due to the urbanization and industrialization, which leads to the global electricity demand. Okay, that's why in many thermal power stations they are generating n number of like huge number of uh, coal fly ash and bottom ash waste as a byproduct. After generating the electricity, these waste that have been accumulated into the soil and deposited and spread towards the global, and these which will be going to create a contamination. Okay, in India, in our country in 22-23, approximately 180 million tons of fly ash that have been produced in annually. These projections, which is rising with the increasing approximately around 2 million tons in upcoming years. See, like in 2024, it have been used in a 180 million. In upcoming years, maybe that have been is an 0.5 fold of enhancement in upcoming years, means the global will be going to uh, get contaminated. We can see in visualization. See, specifically, we have chosen an option is called as in coal fly ash, which is a residue from the coal consumption, which possess a various kind of environmental hazards, which affects the soil health, water quality, crop yield. But we cannot say this is a contaminating product. But see, in one more side, coal fly ash, which contains a good number of minerals. Okay, and we decided due to and based on the literature survey and based on the knowledge we gained from the publications and research articles, we hypothesized that conversion of coal fly ash into nanoparticles, specifically sodium silicate, because these coal fly ash which contains the higher amount of the silicon particle. Okay, for reducing, if we can reduce these kind of particles, means these particles will be used as a biostimulant products in agricultural aspect. That may be either to enhance the growth or reducing the abiotic stress or the biotic stress. Okay, for enhancing the sustainable waste management, this is very, very, very crucial in nowadays. Okay, based on these knowledge and introduction, we have decided aim. this current aim of the study, bridging gap between like waste valorization and agricultural stress management by providing eco-friendly and scalable strategy for enhancing crop productivity by reducing the environmental hazards associating with the coal fly ash disposal. Okay, that's why we aimed like a reduction of environmental pollution and enhancement of agricultural yield by the conversion of coal fly ash into nanoparticles. See, we framed some kind of objectives, like 4 different objectives we have framed. The first one, we synthesized. We synthesized a coal fly ash into sodium silicate nanoparticles. Then we have performed the experiment, like a pod trial experiment, to evaluate the effect of coal fly ash derived sodium silicate nanoparticles on the growth and physiological parameters of rice. Why we have chosen specifically rice means in India, rice is a staple crop. In southern part of India, rice is consumed a meal which fulfilled with the rice. Morning breakfast, afternoon lunch, evening dinner. 3 times we are consuming rice as a very important food. That's why my research crop is rice under salinity stressed conditions. Okay, the third objective, which belongs to investigate the role of coal fly has derived sodium silicate nanoparticle, which reducing the oxidative stress in rice, and followed by the homeostasis level, like ionic homeostasis level assessment in the control and stressed plant with various tissues like shoot tissue and followed by the root tissue. And consider the results what we have analyzed in the precise experiment. See, nothing but we have isolated, we collected some coal fly ash from the reputed contaminated areas, like we have collected these samples from the Tuticorin thermal power station, which is located in Tamil Nadu, India. We collected the sample and we collected the sample. Then we have initially transported via the clear sterile bags into the laboratory conditions, and we have performed the conversion of coal fly ash into a nanoparticle with the help of alkaline extraction protocol with sodium hydroxide. 1:1.5 percentage of the NaOH that have been mixed with the coal fly ash of around 20 to 30 minutes of 200 degrees Celsius. Then we collected jelly-like structure. Then we have centrifuged around 15,000 RPM for 10 minutes. Then immediately the samples were subjected to the calcination of around 600 degrees Celsius for 30 minutes. Then immediately the samples were crushed and then used for the further experimentation. These substances, which will be termed as sodium silicate nanoparticles, for the further experimentation. First, initially we have done some characterization experiment, like because we have converted a large particles to the nanoparticles, we have performed various experiments like UV visible spectroscopy, FTIR analysis, and electron microscopy. See, the UV visible spectroscopy of around 260 nanometer, the ahead peak was absorbed, comes under the FTIR. In FTIR, like the peak in between 2200 to 2000, the specific peak which show a sodium silicate, oxygen, and sodium SiO-Si bond stretch. Okay, which belongs to the specific what we have decided, what we have synthesize the product which contained the silicon as a core metal. Then furtherly, we evaluated with the scanning electron microscopy. See, the first image of SEM analysis is in control, like a sample which showing a spherical and circular in shape of the precise nanoparticles. Then we came to know we have synthesized a specific particles which belongs to all the characterization of the nanoparticles aspect. Then after that, we have conducted an experiment like pot trial experiment. Initially, the seeds, IR64, this rice seed variety was purchased, was collected from Indian Institute of Agricultural Research, Delhi, hosted by Indian Council for Agriculture Research of Ministry of Agriculture and Farmer Welfare Department, and the seeds were mm, sown in the soil that have been filled with the commercial soil, all the nutritional aspect and all the things that have been well known. We were purchased from the online platform, and the first image, and then the specifically, sodium silicate nanoparticles were subjected into 3 days one interval of 4 times, and in between the 100 millimolar of salt stress, like sodium chloride as given as in salt stress. See the image that we have analyzed the good results. We came to know the sodium silicate nanoparticles derived from the coal fly ash, showing an enhanced morphological features. See the image first is in control, and the second image which showing the sodium salt stress alone, like 100 millimolar of salt stress. The image number 3, which is represented like a 25 mg per liter concentration of sodium silicate nanoparticle that was derived from the coal fly ash. And then the fourth one, the 50 mg, like the two concentration we have performed like 25 mg and 50 mg. And consider the fifth image, which is represented the 25 mg of coal fly ash that are specifically given as an 100 millimolar of salt stress, and 100 millimolar of salt stress, along with the fifth last image, which is represented like 50 mg plus 100 millimolar of salt. See, in this image, like we came to know of 1 month of treatment, like a 25th day of analysis, like 0th day to, and then 25th day we have given the treatment of the 5th day to 25th, 20th day, we gave around 4 treatment at 3 days interval, and finally 25th day of treatment, 25th day of sowing, we have analyzed, and we have performed various kind of experiments. See, initially we performed some kind of growth parameters. See, shoot length, we have analyzed shoot length, root length, uh, plant fresh weight. See, you can in that results, you can see the enhancement of growth at the concentration of 25 mg per liter concentration of sodium silicate nanoparticles, show enhancing the shoot length, root length, plant fresh weight, than comparatively reducing 50 mg. Then they were combined with the stress. See, in this experiment, we came to know 25 mg sodium silicate, sodium coal fly has derived with the sodium silicate and enhancing the growth than salt stress treatment, followed by the control treatment. Then we subjected into various physiological experiments like total chlorophyll content and carotenoid content analysis. In this results, like 25 mg coal fly has derived sodium silicate nanoparticle, enhancing improving the precise chlorophyll content, followed by the carotenoid contents. Thereby, in other ants like 50 mg was reduced, followed by the other treatments also reduced, because silicate is a very good metal ion which enhances the chlorophyll properties. Okay. And consider the oxidative stress marker. We have performed some kind of marker analysis like LPO and H2O2. See, this is the markers for salinity, not only the salinity, all the stressed conditions. See, specifically the results like 25 mg sodium silicate nanoparticle treated plants and 50 mg treated plants reduced the markers like LPO and then H2O2, because these kind of enzymes, these nanoparticles which contain sodium and other metals, which reduces the membrane, which increases the membrane permeability and reducing the ROS generations. And we have performed antioxidant enzymes. SOD and POD also analyzed, and enhanced SOD antioxidant enzymes were detected in 25 mg and 50 mg cold fly has derived samples. And we have analyzed some kind of correlation analysis. You can visualize how the correlation, like growth parameter, physiological parameters, and biochemical parameters that are interconnected with each other. See, root length that are interconnected with the shoot length, chlorophyll content that are interconnected with the shoot fresh weight, shoot length, root length, plant fresh weight. And see, LPO and H2O2, that is, whenever the results will be going to reduce, that will be going to opposite towards our precise experiment. See, negatively correlating with these kind of aspect. And comes under the, we analyzed like ion homeostasis. See, we have performed sodium, chloride, potassium, and silicate Si ions with shoot along and root individual parts. See, specifically like salt treated plants alone. See, SS100 and 25 mg of 25 mg nanoparticles plus salt stress, 50 mg plus 50 mg nanoparticle treated plants also higher like sodium content. Comes under the nanoparticle alone treated plants was reduced. Comes under chloride ion also, and then K plus. See, in K plus, like potassium ions like treated conditions in nanoparticle treated like 25 mg and 50 mg, showing the enhanced activity of potassium ions also in the precise shoot along with the roots. This is results, interesting results what we have analyzed. And comes under, this is a graphical illustrations, like we have collected the sample. These samples which will be going to accumulated into the soil, which will be going to create a land pollution, specifically soil pollution, not only soil pollution, water pollutions and environmental pollutions. And we have converted into the sodium nanoparticle, and we have characterized using various kind of various kind of analysis like UV specific and then FTIR SIM analysis. Then we have treated into the plants like rice plants, and rice plants showing enhanced morphology, enhanced photosynthetic pigments, reduced oxidative stress damage, and enhancing the antioxidant capacity. Followed by, we came to conclusion like these sodium silicate nanoparticles showing enhanced rice growth and photosynthetic pigments in plants, and nanoparticles that also reducing the LPO and H2O2 and also enhancing the antioxidant, enzymatic antioxidant properties. And
on how developmental stages and soil factors affect the purple passion fruit metabolome. As a brief introduction, Passiflora edulis form edulis, known as purple passion fruit or in Spanish as gulupa, is a climate native plant to the Andean region belonging to the Passifloraceae family. In recent years, there has been growing interest in its physicochemical profiles, particularly due to the presence of bioactive metabolites such as polyphenols, carotenoids, and tocopherols, which are associated with antioxidant, anti-inflammatory, and cardioprotective properties. Among these metabolites, epicatechin, tocopherols, and beta-carotene are key antioxidant compounds that accumulate throughout fruit development and ripening, and their concentrations can vary significantly depending on the developmental stage. That's why we focused on these 5 metabolites: epicatechin, alpha-tocopherol, delta-tocopherol, and gamma-tocopherol, and beta-carotene due to their antioxidant and nutritional relevance. Despite its importance, there is limited understanding of how these metabolites change during fruit development, how elicitators such as methyljasmonate affect their accumulation, and how soil properties influence them. Therefore, the objectives of this study were evaluate changes in the concentration of target metabolites using 3 approaches. First, during ripening, in immature and mature fruits developmental stages. Second, to assess the effect of methyl jasmonate. And third, the relationship between soil physicochemical factors and metabolites concentrations. Well, this project was developed in a tropical country, Colombia, located in South America, more particularly in an agricultural community which is known for its high production of purple passion fruit on the Indian region. In the municipality of La Unión, the fruits were collected from farms 1, 2, and 3 on the same dates. So the experimental design had 4 sampling dates. In each one, fruits were collected at 2 developmental stages. That is immature, that is unripe green fruit at sampling time, and mature when ripe fruits are ready to eat at sampling time. Next, methyl jasmonate foliar spray treatments were applied, each concentration in triplicate. It means 3 plants per concentration treatments plus 3 plants for the controls. Then the extraction was made in the laboratory with the whole fruit, including the peel. After that, the samples were analyzed by high-performance liquid chromatography with fluorescence detector to epicatechin and tocopherols, and diode array detector to beta-carotene. It's important to mention that the analytical method used was validated based on the parameters of linearity, trueness, precision, robustness, limit of quantification, uncertainty, and matrix effect. Okay. In sampling 0, triplicate soil samples were collected from each farm to determine the soil physicochemical factors. Then that analysis was performed with RStudio using a non-parametric model with a significance level of 0.05. Even the correlation between target functional metabolites and soil physicochemical factors. Well, now let's move into the results. Findings to a stage of fruits comparing in mature show in green boxes and mature in purple boxes reveal statistically significant, which is more visible in the box plots for alpha tocopherol in plot A and gamma tocopherol in plot B. This result will show an increase in the concentration of tocopherols were to be expected, given that the biosynthesis of these compounds is linked to the physiological process of fruit ripening. We also found differences in epicatechin and alpha tocopherol concentration among samplings highlighted in yellow. However, these differences don't directly reflect the impact of fruit developmental stages on the concentration of the functional metabolites evaluated, which is the aim of this research. Moving into the objective 2, findings showed no statistically significance among methyl jasmonate treatments in any metabolite, which could have limited the elicitor effectiveness due to intrinsic physiological factors like developmental stage, anatomical factors like cuticle in leaves, and environmental factors like wind and precipitation, in addition to the concentration of methyl jasmonate applied and the spray volume. Additionally, developmental cues carefully regulate secondary metabolite pathways, including those linked to the synthesis of antioxidants. Mature fruits may have downregulated important biosynthetic enzymes, which reduce the likelihood of illicit or induced stimulation. This is crucial for compounds like tocopherols, whose synthesis occurs in plastids and may decrease as chloroplasts change into chromoplasts during ripening. Therefore, late-stage administration of methyl jasmonate may not cause significant metabolic changes, even if the elicitor is adequately absorbed. Then, when taken as a whole, these findings suggest that the lack of a significant methyl jasmonate effect in our study most likely stem from a combination of environmental limitation, surface-related barriers associated with the curicule, and intrinsic physiological limits related to the fruit development. However, variation were seen among farms in plot A for concentration of epicatechin highlighted in blue, and in yellow among samplings for epicatechin in plot B, and all tocopherol isomers, alpha tocopherol in plot C, delta tocopherol in plot D, and gamma tocopherol in plot E. Despite these differences, don't directly reflect the effect of methyl jasmonate on purple passion fruit metabolome, which is the aim of this study. Addressing objective 3, principal component analysis from Spearman correlation revealed consistent association between soil properties and bioactive compounds across fruit ripening stages. In immature fruits, in plot A, alpha tocopherol was positively associated with available phosphorus and several metals, suggesting that nutrient availability and trace element composition influence early antioxidant accumulation. Epicatechin was linked to soil temperature and specific elements as cadmium and selenium, while gamma tocopherol was associated with cation exchange capacity and total nitrogen, highlighting the role of soil fertility. And in mature fruits, in plot B, beta-carotene remained associated with phosphorus and iron, reinforcing the importance of nutrient balance for carotenoid accumulation. Tocopherols and epicatechin were related, especially delta tocopherol and gamma tocopherol, and epicatechin were related to soil moisture and selected elements as potassium, and lead, indicating that soil conditions continue to modulate the antioxidant profile during ripening. Across both stages, immature and mature, pH show no positive association with epicatechin, suggesting that acid condition may limit phenolic accumulation, as has been reported in other crops. You also see in plot B, pH condition and epicatechin. Regarding to the loamy sandy soil texture, its high drainage capacity may hinder nutrient retention, Also, it promotes the trace element mobility and influence their uptake by plants. This could be reflected in variability in the availability of micronutrients, which affects metabolite synthesis, and the potential accumulation of toxic elements such as lead and arsenic in the fruit. Overall, these findings suggest that soil management practices are critical to optimize the nutritional composition and functional quality of purple passion fruit while minimizing potential risk associated with the metal accumulation. Based on data, we may conclude that this work provides one of the first report linking soil composition with the accumulation of bioactive compounds in Passiflora edulis fruits. Pre-harvest application of methyl jasmonate didn't significantly influence in metabolites level, likely from a combination of intrinsic physiological limits related to fruit development stages, anatomical barriers on the surface of plants, environmental limitation, and some factors in experimental design. The results show that fruit ripening is a key factor, with mature purple passion fruit showing higher levels of antioxidant compounds, especially alpha-tocopherol and gamma-tocopherol, highlighting their enhanced nutritional and functional value. These findings contribute to understanding the coordinated regulation of antioxidant metabolism during ripening and offer practical insights for improving cultivation, harvest, and post-harvest practices. About perspectives, future research should explore alternative elicitors, treatment timings, concentration, and volumes, or storage conditions to optimize the accumulation of bioactive compounds for post-harvest, whereas understanding the genetic and biochemical regulation and the impact of the environment in these pathways into Passiflora edulis.
Sementes de feijão calpi, que é o Vicna, e fizemos 7 tratamentos. Então, o tratamento controle, que não tinha nenhuma inoculação, o tratamento apenas com Bradyrhizobium japônico, na dose de 150 ml para 50 kg de sementes, o tratamento apenas com o Azospirillum brasilense, também 150 ml para cada 50 kg de sementes, e 4 tratamentos de co-inoculação, ou seja, com as duas bactérias juntas. Então, a primeira foi a dose de 75 ml de cada uma das bactérias, o segundo tratamento de co-inoculação, 150 ml de cada bactéria, o terceiro, 225 ml de cada bactéria, e o último tratamento de co-inoculação, 300 ml de cada bactéria. Tudo isso para 50 kg de sementes. Essas sementes foram tratadas momentos antes da semeadura, e aí elas foram semeadas no campo. Com 30 dias após o plantio, nós avaliamos a nodulação, então o número de nódulos e a massa seca desses nódulos, e fizemos também a avaliação de massa seca de raiz e massa seca de parte aérea. Posteriormente, no momento da colheita, nós fizemos a avaliação da massa de 100 sementes e da produtividade. Depois nós fizemos a análise estatística, análise de variância, usamos o teste de Scott-Knott e a análise de componentes principais. Então, aqui nos nossos resultados, nós vemos que a dose de 75 ml favoreceu todos os nossos parâmetros, né? Então, para número de nódulos, a dose de 75, 150 e 225 da co-inoculação foram os melhores para número de nódulos, né? Deram os maiores valores. Para massa seca de nódulos, o tratamento de co-inoculação com 75 ml e 150 mostraram os melhores resultados também. Já para massa seca da parte aérea, novamente a dose de 75 e de 150 da co-inoculação foram os melhores. Na massa seca de raiz, também a co-inoculação com 75, 150 e a dose de 225 foram as mais promissoras. E na massa de 100 grãos, também as doses de 75 e 150, aliás, apenas de 75, e a produtividade também. Então, a dose de co-inoculação de 75 ml de cada favoreceu, né, tanto a massa de 100 grãos quanto a produtividade. E essa produtividade ela foi 64% maior do que o tratamento testemunha, do que o nosso controle, e foi um valor de produtividade 54% a mais do que a média nacional brasileira. Aqui na figura 2 nós temos a análise de componentes principais, mostrando então que todos os vetores das variáveis analisadas, elas estão muito próximas à dose de 75, 150 e 225 da co-inoculação, mas convergindo mais próximo para a dose de 75. E na figura 3, que é uma distância euclidiana, nós temos em destaque essa dose de co-inoculação de 75 ml de cada, que é o quadrado azul, estando mais distante da testemunha, que é o controle. Ou seja, esse tratamento é o que mais está distante da testemunha, no qual nós não usamos nada. Então, para nós, como conclusão deste trabalho, a dose de 75 ml da co-inoculação, né, foi a mais benéfica. Então, utilizando essas duas bactérias, a co-inoculação para essa espécie, para a Vigna, é muito promissora, aumentou muito a produtividade, principalmente em relação à média nacional brasileira, que é baixa. Então, nós conseguimos aumentar a produtividade e uma dose que não é tão alta. Então, 75 ml de cada bactéria é menos ainda do que quando utiliza as bactérias isoladas, né? Então, nós tivemos resultados muito promissores com esse trabalho, mostrando que a co-inoculação é uma prática que pode sim ser incrementada para avaliar, né, para multiplicar o rendimento das culturas. Então, esse foi o nosso trabalho. Temos outros trabalhos com co-inoculação também, outras culturas, principalmente soja. Então, nós estamos trabalhando um pouco com co-inoculação já tem um tempo e vendo que essa prática é muito promissora para aumentar a produtividade, principalmente das leguminosas. Bom, meu trabalho é esse. Se houver alguma dúvida, alguma pergunta, eu possa responder, estou à disposição. Muito obrigada.
Study, we follow these methods. First, the raw material was collected, and then we produced the biochar from this raw material, and then we prepare the substrate. We have 3 substrates. One is cocopeat, one is perlite, and one is biochar / perlite. And then we cultivated the plants, tomato and peppers. For the sampling, at the end of the cultivation period, rhizosphere and bulk substrate was collected, and when we extracted the DNA, we sent this DNA for the RNA sequencing, and we classified the PGPR Foxstov bacteria, and we also perform ecoplate analysis to understand the functional activities of these bacteria, and we analyze these data. In the results section, we saw that the average color development was different between the source groups and between the substrates and roots. And also we compared the PGPR characteristic of these different substrates, biochar perlite, and cocopeat. And this is the heat map of this analysis between them. So as a conclusion, the biochar-based substrate supported the diverse and potentially beneficial microbial communities enriching PGPR-associated taxa, because our objective was to develop a biochar-based substrate from greenhouse biomass waste and to evaluate its effect on plant growth in tomato and peppers, and to assess rhizosphere microbial community structure using RNA sequencing, and to evaluate microbial functional activity and carbon utilization patterns using ecoplate assay to complete the performance of this substrate with commercial perlite and cocopeat. And so as a conclusion, this approach promotes plant productivity while contributing to the microbiome health and environmental sustainability. So we can say that biochar / perlite can be used as a plant growing substrate. And this is my presentation is finished. Is there any questions? Study. So in this study, we collected the raw material from the tomato greenhouse residues, like stems, leaves, and post-harvest biomass was collected and for the biochar production. We produced the biochar. Biomass residues was dried and subjected to gasification under limited oxygen conditions to produce biochar. And then we prepared the substrate. Biochar was sieved and mixed with peat-based compost to prepare the biochar-based substrate. And then we cultivate the plants. Tomato and pepper was grown in a greenhouse under the same conditions. And then we, at the end of the cultivation period, rhizosphere and bulk substrate was collected aseptically. After that, total DNA was extracted from rhizosphere and substrate samples using a commercial soil DNA extraction kit. And then we performed the RNA sequencing on 16S RNA gene amplification and Sanger sequencing for taxonomic identification. And then we classify these bacteria for their functional groups by focusing the PGPR characteristic. Taxonomic data was annotated and screened for plant growth promoting rhizobacteria, as we know as PGPR. After that, community level physiological profiling was performed to assess metabolic activity and carbon source utilization patterns using ecoplate analysis. And then we analyze these data by checking the alpha diversity, richness, carbon utilization profiles were calculated and compared among substrates. So as a result, we know that the average color development values of substrate and root associated microbial communities across different carbon source atoms obtained from the biomass, biolog ecoplate. And we saw that there's not any big differences between these substrate and root across the different substrates. And when we compared the PGPR characteristic of these bacterial taxa identified in biochar, cocopeat, and perlite, we saw that in the biochar perlite composition, the PGPR characteristic of the bacterial taxa was increased, and this substrate promotes the PGPR characteristic of microbial communities. So as a conclusion, the biochar-based substrate supported diverse and potentially beneficial microbial communities enriching PGPR-associated taxa, because our objective was to develop a biochar-based substrate from greenhouse biomass waste and to evaluate its effect on plant growth in tomato and peppers, to assess rhizosphere microbial community structure using 16S RNA gene sequencing, to evaluate microbial functional activity and carbon utilization patterns using biolog ecoplate assays, and also to compare the performance of biochar-based substrate with commercial perlite and cocopeat. So as a conclusion, this approach promotes plant growing while contributing to microbial health and environmental sustainability. Thank you for listening to me.
Project is to develop a system that face and enhance the food security issues. Likewise, making less burden on synthetic fertilizer and incorporating the organic fertilizers to enhance the soil health as well with the passage of time. So here is the outline of my research. So first of all, maize ranked third major crop in Pakistan, and the area of cultivation is 1.41 million hectares, and the production we get is 8.46 million tons in all over the Pakistan. So I'm taking a legume crop, that is soybean, and it is important oil seed crop worldwide owing to its different crop diversity, and the protein content in this is 36 to 40%, while there is a crop that has high oil content as well, that is 18 to 22%. So the problem statement of my research is to differentiate between a monoculture crop or monoculture system that is basically a diversified system and in Pakistan, and most of the farmers are taking this system all over the year. Likewise, it has so many different things and disadvantages and as well as advantages. So here I point some, like the soil-borne diseases in a monoculture system where we only grow one crop at a time in the field. Likewise, it also causes soil erosion, because as a Pakistani, we consume a wheat crop, maize crop, and rice crop, and the cotton, and all are exhaustive crops, taking so much nutrient from the soil and leaving nothing in it. So the soil erosion is a major cause in this system. Likewise, nutrient use efficiency is reducing day by day, because as I proceed earlier, that most of the nutrients are for crops are taking the nutrients from the soil as well as the synthetic fertilizer we are applied in the soil. So adding nothing into the soil, because synthetic fertilizers are mobile or immobile in the soil, that's why. Likewise, water use efficiency is also reducing, likewise the biodiversity, because there is nothing adding in that soil or in that monoculture system. So that's why biodiversity is also reducing in this system. Soil functional microorganisms are decaying day by day, and it ultimately leaves nothing in the soil. As I also mentioned in this presentation, that Pakistani soil has less than 1% of the organic matter. That is likely nothing as it is. So that's why to enhance the organic matter in the soil and to make our soil strengthen, in the case of nutrients, we have to add something into the soil. Likewise, like we can add an organic matter, manures, or we can add an extra crop that can add something or some nutrients into the soil. For example, we can add a legume crop, and adding a crop in a major crop is basically a system that I am defending right now, is the intercropping system. So intercropping system is some word also defined as diversified cropping system, that not only reduce the soil-borne diseases, but also reduce the soil erosion, because two of the crop taking the same piece of land as well as taking the same resources and strengthen the soil particles into it, intact the soil particles, and that's why the soil erosion is reducing in this diversified crop system. Likewise, nutrient use efficiency is enhancing because most of the legume crops, like a soybean crop, add atmospheric nitrogen into that soil, and that ultimately not only enhance the soil health in a case of nutrients, but also increase the nutrient use efficiency for the other crop, like as exotic crops, as I mentioned, like maize crop. Likewise, the biodiversity and soil functional microorganisms also enhance in that soil, because the diversified soil root system also make the soil more favorable conditions to the microorganisms to grow in that conditions. So the possible solution for this is the intercropping system, and here is some pictorial view that I mentioned in this slide. For example, this is the maize crop we are adding. Likewise, these two crops are soybean plants, and then again the maize crop. So this intercropping system is showing 2:2 ratio crop. Okay, okay, and as we know that maize to maize plant distance is 40 centimeter, and we're taking the maize to soybean plant distance is 60 centimeter. Likewise, again 40 centimeter, 60, and then again 40. Whereas in the monoculture system, we only grow legume crop or the other crop with the same distance. So we have a space in these two plants, we can grow a different and other plant in the system. So what is the cereal legume intercropping system, and how we can defend this system into and develop this system and adapt this system into our environment? So cereal-based cropping systems, especially like maize, sorghum, and millet, offer promises for intercropping of soybean, owing to its flexibility of planting geometry and compatibility of growing season. Because soybean crop in Pakistan has two major seasons, like we also grow season as well as in the July season, and we have two major seasons to grow and incorporate this legume crop into our major crop. Likewise, we can add into maize crop or the soybean or the millets as well. Likewise, intercropping legume with cereals may be a mean of increasing soil nitrogen efficiency. As I mentioned earlier, that legume crop has the capacity to convert the atmospheric nitrogen into the edible form of the plant. Likewise, intercropping of soybean enhanced nutrient like NPK uptake of maize by 8 to 10%, and that is held research by Rukhsar et al. At 2020. So this is the concept of intercropping system that is happening above and underground. For example, we have the atmospheric nitrogen system that is fixed by the legume crop roots that have the nodules in it, and these nodules have biological nitrogen fixing bacteria that is called as rhizobia. These rhizobia not only convert the atmospheric nitrogen into the available or the nitrogen fixation form, so that the other plant, like the compatible plant or the companion plant like maize, can take this nitrogen. So this not only increase the efficiency of the nitrogen system in the maize crop, but also make a less burden on the farmer to add the synthetic fertilizers from the above ground. Likewise, if these legume crops have these leaves deteriorate into the soil, they can also add a crop residue, and hence the nitrogen in these leaves ultimately formed a mineral nitrogen that can be used by the companion crop. So the objective of this project is to quantify not only the nitrogen use efficiency of maize soybean intercropping, because these both crops have different nitrogen use patterns. Through integrated nitrogen management for enhancing not only the farm productivity but also the sustainability of the system, because our ultimate or a long-term goal is to make our soil healthy as well as sustainable in a sustainable form. So these are a design of my project where we use the plot area, and the location is Agronomic Research Farm, University of Agriculture, Faisalabad. Okay, so these majorly project has two different factors. In factor one, we take the sol maize, sol soybean, and maize soybean intercropping, and these are the major plots. While we also divide this major plot into subplots, and these subplots has nitrogen source factor, where T1 represents the recommended nitrogen dose for both the crops. Like if at the sole crop, we adding a maize nitrogen recommendation that is 210 kg per hectare. Likewise, the soybean nitrogen recommended nitrogen that is 30 kg per hectare. So we adding these adding these nitrogen sources separately in both the crops. Likewise, T2 representing the 75%, the reduction of 25% in the recommended nitrogen dose, and adding the 25% compost to make the nitrogen availability from this compost and making 100% overall. Likewise, T3 reduction of 50% of recommended nitrogen dose, that is from a synthetic fertilizer source, as well as adding the 50% of the compost to make this compatibly equals to 100. Likewise, T4 reducing further 25% recommended dose, as well as adding 75% compost with it. And in the T5, adding totally 100% compost or the organic fertilizer and having nothing included synthetic fertilizer in this system. These are some yield parameters that I taken from maize crop as well as from the soybean crop to measure for both in the sole maize crop as well as when the maize is grown with the soybean. Likewise, in the soybean yield parameter also indicating these all the parameters are taken from the sole soybean. Likewise, also from the soybean that was intercropped with maize crop. So this is the pictorial or the graphical view where because I mentioned that I have 2 year study in this project, that is 2022 and 2023. So the grain yield in tons per hectare show a drastic change in both the year. Like as you see, the light green shade shows the sole maize cropping, whereas the darker green shows the maize when it is intercropped with soybean. This yield basically, or that this bar is not showing both the maize and the soybean collectively. This only shows the maize grain yield when intercropped with soybean. So it is subtracted from the soybean yield. So you can see that when there is a sole crop, we have a higher grain yield, whereas as you see in the intercropping system, a minor change shows in the grain yield of the soybean crop in 100% recommended nitrogen dose. Whereas if you can see that during 75% recommended nitrogen dose and adding a 25% of organic fertilizer into that treatment, we see that not only the grain yield somewhat increasing in sole, but also in the intercropping system as well. It means that both these, both maize when grown in the sole or in the intercropping system, having a high grain yield production as and reduce nothing in it. Likewise, in the 2023, there is similar ranges in the next year. Likewise, the last bar shows the 100% compost level, which shows that if you can add 100% compost in the first year, the crop cannot take the nitrogen because this is not well decomposed into the soil and available to the plant in the first year. While if you see in the next year, 2023, the same land, when we add the compost again, the last year, the subsequent previous year compost adding somewhat nitrogen, release somewhat nitrogen from that system and add into the soil, that also taken from this by maize in both sole and maize crop. So you can also conclude that while year-wise adding the compost into the soil, you can have major change with the passage of time, like within 2 to 3 years, or maybe with after 10 to 15 years. Likewise, if you can see the biological yield that also measured in ton per hectares, you can see that the intercropping bars are more as compared to the sole maize crop, which is shows that the biomass yield is increased when maize crop is grown with the soybean. That only possible because the nitrogen itself enhance the biomass of the crop, and as soybean adding nitrogen, atmospheric nitrogen into that soil and taken up by the maize crop as well. So that biological yield enhances in this system. Likewise, in the second year, also the biological yield of the intercrop maize is in more as compared to the sole crop. So here is the soybean yield when grown sole or when grown with the maize crop. So you can see that the grain yield is 2.5 tons per hectare when 100% recommended nitrogen dose of the soybean adding into the plot. whereas the intercrop has a less yield in it. This is because of the shading effect of the maize crop. As we all know that maize is a fast growing crop and it covers the soybean crop and shade when the soybean is in the flowering stage. So that's why during the flowering stage, soybean cannot take most of the sunlight from the above because of the shade of the maize crop. So that's why somewhat of the grain yield is reduced in the soybean. But in the sol crop, we can get 2.5 tons, 4 tons per hectare. Likewise, this is the same pattern in the 2023 as well. Whereas the biological yield of the soybean is also enhanced in the sol as compared to the intercrop system. So this is the same pattern as grain yield or in the biological yield. Here are some intercropping indices where we can calculate how much intercropping is efficient for our system or not, or the combination of the two crops are beneficial for us or not. So we can conclude these things from different intercropping indices, like the land equivalent ratio, which shows that if a piece of land, like for example, 1 hectare of the land is compared by sol maize crop, whereas if we taken the same 1 hectare land and we grow maize as well as the soybean into the same land, how intercropping enhance that land use efficiency. So basically the blue bar showing the more than 1 LER, land equivalent ratio value, which shows that LER is beneficial for maize and soybean intercropping system. So that's why this combination is beneficial. Likewise, during the subsequent year 2023, intercropping system is also beneficial in this way. Whereas you can see that if we grow only a sol maize or the sol soybean, the values are less than 1. So the land use efficiency in the sol cropping is not beneficial as compared to the intercrop system. Similarly, if we see that 100% recommended nitrogen dose adding or the 75% nitrogen dose plus 25% compost shows the more advanced shows as compared to the 100% compost or the 50% of both the recommended nitrogen dose and 75% compost. So this is the drastic change we can see in a sense of nitrogen resources or the nitrogen combinations, likewise in the sense of intercropping system. So the next one is the competitive ratio. Like this shows that how much maize is more competitive or the soybean is competitive towards each other or the companion crops. So the blue bar is basically a maize competitive ratio. So this shows that maize is more competitive as compared to the soybean in a sense of taking nutrients from the soil as well as taking water from the soil, likewise the taking radiations or solar radiations from the sun. So maize is more competitive as compared to the soybean. And we also want this pattern because maize is our main crop and we cannot afford any reduction in the yield or the biological yield of the maize crop. Likewise, we can somewhat taken soybean for granted because it is just an intercrop that we can add it in that combination. So the 100% recommended nitrogen dose and as well as 75% nitrogen and 25% compost shows more competitive results as compared to the other treatments. Likewise, in the next year 2023, 75% nitrogen and 25% compost shows more competitive results as compared to the competitive ratio of the soybean crop. So here is the monetary advantage index, which shows that how much money we can get while in taking intercropping system in different nitrogen combinations. You can see that the line is more inclined towards 75% nitrogen and 25% compost in compared to 100% recommended nitrogen dose. This completely shows that if I can reduce the nitrogen from synthetic fertilizers, we can not only enhance the maize yield, but also make a healthy environment for the soybean also to develop this system. Because we know that soybean has less recommended nitrogen dose as compared to the maize one. So that's why. But totally the 100% compost adding shows minimum results, or the loss towards the monetary advantage index at the end of the year. Likewise, 2023 also shows the same result, where 100% nitrogen recommended nitrogen dose also shows the best result, as well as 75% nitrogen plus 25% compost also shows the similar or somewhat similar results in the system. So ultimately, the most beneficial or the most competitive one for us is the system productivity index, because ultimately we have to focus on our system, on our farm productivity, as well as on the soil sustainability. So you can see that the line is more inclined towards 75% recommended nitrogen dose from the synthetic source, likewise 25% from the organic source, and the similar results are shown in the 2023, which shows that the addition of organic matter not only enhance the system productivity, but also enhance the soil sustainability and soil health with the passage of time. Likewise, this also shows that both this combination has more benefits towards maize soybean intercropping system. Whereas only adding 100% compost is not beneficial unless it is beneficial for soil health, but 100% compost during the first or the second or the third year cannot show the best result, because the compost is taking time, like 4 to 5 years or maybe 6 to 7 or 10 years to decompose into the soil and adding their nutrients into the soil. So it takes time. We cannot say that 100% compost can show a best result within 1 to 2 years. That's not possible. So here is the conclusion of my project. The intercropping can lead to higher yield and increased benefits due to complementary growth patterns and efficient resource utilization between two crops. As you can see, both the crops have different canopy geometry, likewise different root system, as soybean is a tap root system, while maize has a shallow root system. So taking the water more efficiently through maize, as well as if the water leaks down from the root zone of the maize crop, this water can be beneficial or uptake by the soybean. So what resource utilization is the best way in the intercropping system. Incorporating intercropping techniques and utilizing a combination of different nitrogen sources such as compost and synthetic fertilizers can be an effective strategy for improving not only crop yield but also soil health and promoting the sustainability in the agriculture. Continued research and field trials are essential to further optimize the nitrogen use selection, because we only optimize 5 different combinations, and in the future we can also optimize further nitrogen combinations or more close results. We can get more close results with this. Application rates and the timing of maximizing farm productivity and sustainability in cereal soybean intercropping system. Here are some references, and thank you so much listening me and taking my work with you guys is really honorable.